They are mainly neutrophils and macrophages. The other role for macrophages is not only cleaning. They also produce cytokines and growth factors, and they kind of prepare an implant surface for mesenchymal stem cells. Mesenchymal stem cells are very important because when they arrive to the implant surface, they have to attach, proliferate, and they have to produce a new bone. They have to connect our implant to the existing bone and produce a new bone. So they have to differentiate into osteoblasts, which are bone cells. At the same time, when this osteo or like hard tissue integration occurs, we have soft tissue integration, and this is in this abutment part. And there we have another cell types that come to the implant site, and these are for epithelial cells, like for example fibroblasts, and their main role is to produce connective tissue. So the success rate of dental implants is approximately 94 to 95%, and you can say it's quite high. So why do we modify the surfaces? Why is it so important? This is because of the complications. So after implantation, we can have peri-implant diseases can occur, and we have two types of them, peri-implant mucositis and peri-implantitis. Both of them are of bacterial origin, and peri-implant mucositis or mucositis, it affects approximately 43% of the patients, and it only affects the gingiva above the implant. However, it can be, we can also have more severe disease, which is peri-implantitis, and it is in approximately 22% of the patients. And here we have not only inflammation at the mucosal level, but bacteria can go inside the bone pocket, and it can bone resorption or bone loss can happen as well. So we can change the surface of the implant by applying two strategies. We can change either topography, it doesn't work. We can change either topography or chemistry of the surface. And when we talk about topographical modifications, so we mainly aim to change the physical structure of this, the physical surface structure. When we talk about the chemistry, we aim to change the surface composition. And we have different methods to do that. For topography, we can, for example, apply sandblasting. It's also called grid blasting. So we have some particles, and by using those particles, we can form this macro roughness, these valleys and peaks that I already showed you. We also have acid etching. I will explain it in details later. And we have also other methods. There are more, like I showed you only examples. With changes in the surface composition, we can also apply many different methods. And today I would like to focus on coating with bioactive materials. We can use very different molecules to coat the implant surface. For example, peptides, extracellular matrix, and other molecules. But what we do in our lab, we work with polyphenols, which is, for example, tannic acid. So polyphenols, it's a huge group of different compounds. They occur like naturally in plants, so you can find them in, for example, blueberries, raspberries, and chocolate, tea, grapes, red wine as well. So it's very healthy to drink red wine. And as I told you, there are like hundreds of them. It has been shown that some polyphenols, for example, quercetin that Manu worked with, it can enhance implant integration both at soft tissue level and bone level, and it also can work antimicrobial. So those kinds of modifications we call multifunctional modifications. In our lab, we work with tannic acid. This is that powder that I work with. And this is the structure of tannic acid molecule. It's very complicated, but I'm showing this structure to you because it's important to show you the structure to understand why tannic acid has so many health beneficial properties. So as you can see, the tannic acid molecule contains many benzene rings and with hydroxyl groups. And because of these hydroxyl groups, tannic acid can interact with many different molecules, with metals, but also with polysaccharides, proteins, and so on. And due to its structure, tannic acid has been shown to have many beneficial effects for us, for our health. It has been shown that it's antimicrobial, antifungal, antioxidant, anti-aging, and all these anti-things. And at the same time, it's biocompatible. So now I will talk about my PhD project and surface modification. So in our lab, we had two guys. They are both German, Sebastian and Florian, and they both developed and optimized tannic acid coating on titanium implant surfaces. So here you have tannic acid solution, and after adding a magical ingredient, which is silicic acid, you can form this very nice nano coating, or coating in nanometer range. And the longer we coat the surface, the thicker coating we have. And for example, here we have very thin coating. When we coat the surface, for example, for 30 minutes, it's approximately 7 nanometers. After some hours, we can have very thick coating, also in nanometer range, but it's like 50, 60 nanometers. So now we go to my project. So we have developed this coating, but we don't know how the human cells and human tissue will respond to the coating. So in the first part of my PhD project, we were focused on soft tissue integration. So I worked with this smooth surface, and we coated the surface with tannic acid. It was a thin coating, 30 minutes deposition, and then we added human gingival fibroblasts, and we wanted to study how they respond to the coating. For that, we employed 3 different methods. It was live cell imaging, QCMD, that I will explain you in detail soon, and immunostaining. So with live cell imaging, we were recording cell behavior over time for approximately 16 hours. So and I will show you some videos. So due to applying live cell imaging, we knew how the cells behave on the surface, how they spread, how they move, whether they were viable or not. Then we employed crystal quartz microbalance with dissipation monitoring method, and we can use this method to monitor the dynamics of a cell attachment, how the cells behave on the surface over time. And at the end, we wanted to also take a look at cell morphology, how they spread on the surface and how strong they are attached to the surface. So first, I would like to explain you what quartz crystal microbalance with dissipation monitoring, or QCMD, is. So here you can see me in the lab working with that method. It seems to be very complicated, and I don't know all the physics behind the QCMD, but I can briefly show you that we work with these quartz sensors. They are coated with titanium, so they imitate the smooth surface of the implant. Then we place this sensor into the QCMD chamber, and then chamber is placed into this machine. And when we apply an alternating electrical voltage, the sensor starts to oscillate. And when we add cells, and when the cells start to deposit or attach to the surface, the frequency changes. So this is the principle of the QCMD. During QCMD experiments, we can monitor changes in two parameters, frequency and dissipation. And the only thing that you have to know is that when we add cells or anything, polymers, other molecules, to our QCMD chamber, when the mass increases, the frequency decreases. We also can monitor changes in dissipation. Dissipation is like energy loss. And when we add, again, cells or other molecules to our QCMD chamber, the dissipation can increase, and then we know that we work with quite soft and viscous layer. But when we work with very rigid layer, then the frequency becomes very low. And QCMD allows us to measure the dynamics of attachment of different molecules in nanometer range. So, for example, like proteins, DNA, antibodies, but also cells, which are very huge. What are advantages? So first of all, the method is cost efficient. It's very cheap to run these experiments. It's label-free. It means that we don't have to use antibodies to, for example, stain cells. We monitor cell behavior or cell attachment in real time, because we monitor in real time changes in frequency and dissipation, and the method is very sensitive. So this is our setup in our lab. We have this QCMD machine that is connected also to our confocal microscopy. So we use two QCMD chambers, these guys. So what I forgot to tell you is that the chambers that we use had this window. So this is why we could use confocal microscopy. We could, by using like an objective, we could observe the cells. So what we did here, we had one QCMD chamber that was in our QCMD machine. The second one was placed under the microscope, and we ran an experiment overnight. Yeah, and what we did, we had this crystal sensor, then we coated it with tannic acid, then we added proteins. So proteins were dissolved, or we used FBS, bovine serum, in different concentrations, ranging from 0% serum-free up to 10%, and then we added fibroblasts. This is typical result from our QCMD method, and it might be very complicated for you, so I'm not going to go into the details, but what you basically see here, this is a plot dissipation versus frequency over time. So here we monitored cells up to 500 minutes. And what you can clearly see from this plot is that cells behave differently in different serum concentrations. So when we increased the serum concentration, we got different QCMD signals, what can mean that we have different cell adhesion dynamics. And here we have some results. I can show you the videos. So these are gingival fibroblasts that were stained with cell tracker. This is a green stain, a green dye. And the red cells should, I mean, you will see some red cells here, and they are stained with propylium iodide. So we can clearly see that cells started to attach and spread very quickly within 30 minutes. They were like totally spread. And they didn't do much after the initial spreading, so they were like mostly immobile on the surface. However, when we add 10% serum, the behavior is totally different. And take a look at this cell. So we can clearly see that cells are not happy. They attach, try to spread, but the surface is not attached, and they are more motile. They are migrate towards like better surface, and you can see that we have some dead cells as well. What we got from live cell imaging, so it's like here you can see cell motility plots, and you can see that in different serum concentrations, the cells were more and more motile. So these lines we tracked like 10 cells per condition, no 40 cells per condition, and we made these plots. So you can see that in serum-free conditions, cells are like almost immobile, then they do not move much. However, in 10% medium, they migrate a lot. So when we increase FBS concentration, the cell motility increases. Then we stained some cells with 2 dyes, like 3 basically. So here you can see gingival fibroblasts that were attached to titanium surfaces coated with tannic acid coating, tannic acid. And here you can see the cells that were cultured in serum-free medium, 0.1%, 1%, and 10%, and we incubated the cells for 1 hour, 2 hours, 6 hours, and 24 hours. The cells were stained with phalloidine to visualize cell cytoskeleton. This is the red color. They were also stained with nuclear DNA, so you can see the nuclei in blue. And we also used a vinculin. Vinculin is a protein that you can find in focal adhesions. Focal adhesions are like contact points that cells make with the underlying substrate, and they are very important for their cascades and all the pathways within the cell. So you can clearly see that in serum-free conditions, the cells started to form these focal adhesions contact points as early as 2 hours after seeding, and the process was delayed for higher serum concentrations. What you also can observe is that in 10% serum, we had mostly these round and spherical cells that were not happy. They didn't want to attach to the surface. After 24 hours, we could barely see some nice elongated cells. So what is the conclusion of the first part of my PhD project is that cell adhesion depends on both protein and layer and the underlying substrate. So here you can clearly see that if we add serum proteins or if we pre-coat our surface with serum proteins, the cell behavior will be different. Why is it so? And why do they attach differently to the surface with increasing serum concentration? We don't know yet, but it might be because of the interaction between proteins and tannic acid. And this is a part of Enrique's project. So the aim of this project is to investigate how proteins bind to our tannic acid surface. What we also tried to do, it was to combine tannic acid coating with another molecules. So what we can do is to form a thick layer that consists of multi-layers of different molecules. In our case, we used polyamino acids, polylysine and polyarginine. So we deposited tannic acid, then a thin layer of polyamino acid, let's say polylysine, then tannic acid, and so on. So we built a very thick multi-layer to check whether it improves cell adhesion. We saw an improvement compared to tannic acid coating, so this can be a solution. But I'm not going to show you any results. It's we are writing the paper now. The second part of my PhD focuses on this part of dental implant, which is a rough surface of the rough surface. And in the second part of my PhD, we aim to improve bone tissue or like hard tissue integration. So what we did in this case, we worked with these titanium discs that imitate dental implant. They had rough surface, and some of the discs were coated with tannic acid, and other ones didn't have any coating. Then we grow bacteria on the surfaces. This is probiotic bacterium Lactobacillus crispatus that we then inactivated, killed, and then we studied, or we still do it, the response of different cells that are involved in hard tissue integration, how our coatings impact integration, and whether our coatings or our surface functionalization has any antimicrobial activity, but I will show you in a minute. So why we have chosen Lactobacillus crispatus to form like a biofilm on our coating? So first of all, this is normal human microbiota. You can find Lactobacillus crispatus mainly in vagina, but it has also been found in oral cavity. So this is physiologically relevant. Then Lactobacillus crispatus shows antimicrobial, like health beneficial effects by killing pathogenic bacteria. It is because it can release different compounds like bacteriocins, hydrogen peroxide, and lactic acid that works antimicrobial. And at the end, Lactobacillus crispatus is able to form a biofilm. To make this study a little bit more complicated, what we did when we had those two surfaces, titanium and tannic acid coated, we split them into two groups. In some of the coins, we added just a little bit of Lactobacillus crispatus because we don't know how much is enough. And on the other surface, other disc, we added plenty of bacteria, so we were able to form very thick coating or like biofilm. You can clearly see it here. So this is a titanium disc where we added just a little bit of bacteria. You can see Lactobacillus crispatus on the surface. However, when we added more bacteria, we got thicker and nicer biofilm. On tannic acid, bacteria behave slightly differently. They were forming this cluster. It can be due to antibacterial effect of tannic acid. However, what surprised us a lot, when we added a lot of bacteria, we got very thick biofilm that even cracked, so we had many layers of bacteria on the surface. The next step was to study how human cells interact with this kind of modifications. So what we did first, we cultured mesenchymal stem cells on our modified surfaces, and we wanted to check whether the surface is toxic for human cells and whether the cells are able to survive within 24 hours. So the first plot shows you the cytotoxicity of the surface that we measured after 24 hours of human cell culture, and the cytotoxicity was below 10%, so we consider this surface not to be toxic for human cells. When we take a look at cell viability, whether the cells survive or not on the surfaces, you can clearly see that on titanium surfaces it was an improvement for the titanium discs coated with Lactobacillus crispatus. For tannic acid, the cell viability was lower compared to control. This is what we usually observe. And we saw an improvement when we coated tannic acid, or when we deposited Lactobacillus crispatus on tannic acid coating. It was a thick biofilm. This is how the cells looked after 24 hours of culture. So we have the same cells, mesenchymal stem cells. Over here, they were stained with phalloidin to visualize cytoskeleton and counter-stained with DAPI. So what you see here, this is not an artifact signal. So here you can basically see bacteria that were stained with DAPI because they contain, of course, DNA and they give a blue signal. This is the whole coin. This is how it looks under the microscope. This is 5x magnification. But what I wanted to show you or emphasize here is that when you take a look at the titanium coins only first, and the highest magnification, you can clearly see that when we sit cells on thick biofilm, the cells are more elongated. They look to be a little bit more happy. On tannic acid, this is the normal view that we see on a thick tannic acid coating. The cells are dying, unfortunately. However, when we add some cells, it looks like the cells, you have, first of all, more cells on the surface, and the cells look better, they are more elongated. The next step in my project was to test the effect of surface functionalization with probiotic bacteria on osteogenic differentiation. And as I told you at the beginning, when we talk about hard tissue integration after implantation, the mesenchymal stem cells are recruited to the implant site. And after some days, they differentiate through preosteoblasts, major osteoblasts to osteocytes, and we call osteoblasts bone-forming cells, while osteocytes are bone remodeling cells, and both of them are very important for implant integration. What you can see here, there are some markers that are produced by cells over time. And what we did, so we prepared our coatings, we seeded mesenchymal stem cells, and then we cultured them for 21 days. And we took a look at different expression of different markers. I'm not going to show you all of them. The only thing that I want to show you is the metabolic activity or viability of the cells. So you can clearly see that after 21 days, we still have live cells on the surfaces. It seems to be that we have a little bit more cells on thin Lactobacillus crispatus biofilm. However, it's not significant, statistically significant, so we cannot say it's better. Then I showed you only one result for the markers. It's ALP, alkaline phosphatase. So this is, this is this enzyme that is produced already by preosteoblasts, then by osteoblasts, and then it's not produced by osteocytes. So it's a marker of early differentiation process. And what you can clearly see at day 3, we had almost like nothing, and the production of ALP increases over time. And after 21 days, it's the amount of ALP is comparable for titanium control and bacteria-coated discs. For tannic acid, here we had like no cells, they died. They don't like tannic acid. However, when we coated tannic acid, or when we formed a biofilm on tannic acid-coated surfaces, it helped and the cells survived and produced ALP. Then we wanted to test antimicrobial properties of our surfaces. So what we did, we had our titanium disc with Lactobacillus crispatus biofilm. Then we added Staphylococcus epidermidis. This is a pathogenic bacterium. On our discs, we cultured them for 12 hours, and we wanted to check whether our surfaces can kill or prevent bacterial invasion. And here you can you have the results. I'm not going to go describe everything, but this is the most important result. This is tannic acid coated coin or disc with thick Lactobacillus crispatus biofilm. So compared to other conditions, you can clearly see that this peak, don't look at this peak, this is something normal, but when you take a look at this peak, it's much lower. It means that Lactobacillus crispatus inhibits Staphylococcus epidermidis growth. The discs that we had after this experiment, we further dehydrated and imaged with electron microscopy. So here you can see Staphylococcus epidermidis, round bacteria. On our titanium control, after 12 hours, you can clearly see that it forms a biofilm. It adheres, like a lot of bacteria adheres to the surface. When we have thin biofilm of Lactobacillus crispatus, it doesn't help. We still have a lot of staphylococci. However, when we have thick biofilm of Lactobacillus crispatus, we have only few colonies of Staphylococcus epidermidis. And this is the same for tannic acid. Tannic acid without any Lactobacillus. Actually, tannic acid, that's supposed to be antimicrobial, it worked as a glue. So we had a lot of Staphylococcus epidermidis here. When we coated with a thin layer of, when we had thin Lactobacillus biofilm, it didn't help. And again, when we had very thick coating or very thick biofilm of our probiotic bacteria, only few Staphylococci colonies we could observe. That's all. Now I want to only briefly show you what I'm going to do here. So I prepared this presentation previous week, and our plans changed a little bit. So I might lie a little bit on the slide. But what was our plan? So what we wanted to do here, we wanted to isolate the monocytes from human blood, then differentiate the monocytes into macrophages. Then we wanted to seed those macrophages on our surfaces that I brought with me from Oslo. And then we want to investigate whether these M0 macrophages differentiate into pro-inflammatory or anti-inflammatory, thank you, anti-inflammatory macrophages. So this is, we want to assess it as I showed you at the beginning. Heart tissue integration involves macrophages. Acknowledgements at the end. I would like to acknowledge my supervisors and my whole group, and this is our Luther team, as Manu mentioned at the beginning. So our Luther project is founded by the Research Council of Norway. It was a huge grant that where we aim to develop tannic acid modified implants for improved wound healing and implant integration. The project is led by my supervisor, Hanna, but we have also some collaborators. This is Professor Fernanda Petersen from our faculty. She's a microbiologist. Here we have Manu. We have also Professor Messerschmidt from US. And we still work on this project. My research space here at IdISBa is founded by UIO Life Science, and that's all. If you have any questions, I will be happy to answer. You can reach me through my email, or you can find me on LinkedIn, and I hope you are not very bored. Thank you so much.